In the name of Allah, who is most beneficent and merciful. My name is Ayman Fakat and my department is BS Chemistry. Today the topic of my presentation is about chloroxylene. These are some main points: introduction, structures, history, properties, chemical properties, physical properties, chemical reactions, uses and side effects. Introduction. Chloroxylenol, also known as para-chlorometaxylenol, Pcmx, is an antiseptic and disinfectant which is used for skin disinfection and together with alcohol for cleaning surgical instruments. It is also used with a number of household disinfectants and wound cleaners. It is thought to act by disrupting microbial cell walls and inactivating cellular enzymes, and is less effective than some other available agents. It is available as a liquid. Structures. There are different interactive chemical structures models that are described below. Ball and sticks, wireframe and space filling. The structure of ball and stick, structure of sticks, structure of wireframe, and last space filling structure. History. Chloroxylenol was first made in 1927. It is one of the World Health Organization's list of essential medicines. It is sold in a number of formulations and under a number of brand names including Dettol. Soon after it was created, was then called Pcmx, but this was thought to be a poor name and it was renamed Dettol. Then in 1932 it was marketed in Britannia and in India. It had white on green bottle with a white sword. Next, chemical properties. Appearance: white or cream crystalline powder. Boiling point 246 degrees Celsius. CAS number 88-04-0. Density 1.183 gram per centimeter cubic. HS code 2908100. IUPAC name 4-chloro-3,5-dimethyl-phenol. Melting point 114 to 116 degrees Celsius. Molar mass 156.61 gram per mole. Molecular formula: carbon 8, hydrogen 9 and CiO. Next, physical properties. Property del G value minus 56.92 unit kilojoule per mole. Next, delta H in the form of gas minus 187.91 unit kilojoule per mole. Delta hydrogen in the form of union 19.72 kilojoule per mole. Delta of hydrogen in evaporation 54.40 value than unit kilojoule per mole. MCV volume 117.930 unit mole per mole. PC 4041.50 and unit K Pascal. Next, chemical reactions. Chloroxylene undergo following reaction. First, dechlorination, dehydrogenation and oxidation. First, benzene first hydroxyl group अटैच है, टू साइड्स पे CH3 एंड 1 क्लोरीन अटैच है. जब इनकी डीक्लोरीनेशन की देन बेंजीन के साथ OH अटैच क्लोरीन ख़त्म हो गई है. नेक्स्ट, डिहाइड्रोजनेशन में OH हाइड्रोक्सिल ग्रुप की जगह डबल बॉन्ड ऑक्सीजन आ गया. नेक्स्ट, अरोमेटिक रिंग ऑक्सीडेशन में HC डबल बॉन्ड OH CH3C डबल बॉन्ड OH C bond C double bond O bond OH. Then last mineralization पे CO2 plus water produce हुए. Next, uses. Chloroxylenol is used in hospitals and households for disinfection and sanitation. It is also commonly used in antibacterial soaps, wound cleansing applications and household antiseptics. Testing has shown products containing chloroxylenol effective against the SARS-CoV-2 virus and orthopox viruses. Side effects. Chloroxylenol is generally slightly to moderately toxic to humans but high toxic for the eyes, causing severe eye irritation. It is practically non-toxic to birds and is moderately toxic to freshwater invertebrates. It is highly toxic to fish, cats, and some amphibians and should not be used around them. It is mild skin irritant and may trigger allergical reactions in some individuals. Next, effects on human. Excessive exposure to chloroxylenol has the potential for causing death. It can be poisonous when and even when it is unintentionally inhaled. A medical study in Hong Kong which analyzed 177 cases of Dettol ingestion that resulted in emergency department treatment, 95% of which were intentional, concluded that Dettol poisoning resulted in serious complications in 7% of patients, including death. Next, effects on animals. Chloroxylenol is toxic to many animals, especially cats. Phenolic compounds are of the particular concern because cats are unable to fully metabolize them. A cat may swallow the product by licking its paws after they have come into contact with it. Australia, chloroxylenol spray has been shown to be lethal to cane toads, an invasive species that was introduced from Hawaii as a result of bad judgment in 1935. It had been hoped that the amphibian would control the cane beetle, but it became highly destructive within the ecosystem. Spraying the disinfectant at close range has been shown to cause rapid death to toads. It is not known whether the toxins are persistent or whether they harm other Australian flora and fauna. Owing to concerns over potential harm to other Australian wildlife species, the use of chloroxylenol as an agent for pest control was banned in Western Australia by the Department of Environment and Conservation in 2011. Next, applications. Pharmacodynamics, protein building, half-life and toxicity. Application first, pharmacodynamics. Chloroxylenol is substituted phenol which has been widely used for many years as an ingredient of antiseptic and disinfectant products intended for external use. It is known to be bacterial in low concentration to a wide range of gram-positive and gram-negative bacteria. Mechanism of action. As a phenol antiseptic, it is believed that the hydroxyl OH groups of the chloroxylenol molecule binds to certain proteins on the cell membrane of bacteria and disrupts the membrane so as to allow the contents of the bacterial cell to leak out. This allows chloroxylenol to enter the bacterial cell to bind further with more proteins and enzymes to disable the cell's functioning. At particularly high concentrations of chloroxylenol, the protein and nucleic acid content of targeted bacterial cells become coagulated and ceases to function, leading to rapid cell death. Absorption. No chloroxylenol was detected in the blood following the dermal administration of 2 g of p-chloroxylenol in an ethanol olive oil vehicle in human subjects. After a dose of 5 gram, only traces were found. After 80 gram, 1 milligram was found in the blood after 3 hours and 4 milligram over dilution L after 24 hours. After a dose of 20 gram, 4 gram was measured after half an hour and 1 milligram was present at 72 hours. For antiseptic purposes, chloroxylenol is considered to be well absorbed when applied to the skin. Volume of disruption. The only data available regarding the volume of disruption of chloroxylenol is the mean of 22.45 L determined after 200 mg intravenous single dose of chloroxylenol was administered to healthy mongrel dog subjects. Protein binding. One study determined the protein binding of chloroxylenol to be approximately 85.2% plus minus 2.32 for serum albumin and 89.8% plus minus 2.99 for whole human serum. This 20% protein binding drug and albumin. Metabolism. Certain animal studies have shown that following dermal applications of chloroxylenol that the absorption was rapid with a Pcmx 1-2 hours and that the administered substance was exerted via the kidney with almost complete elimination within 24 hours. The primary metabolites discovered in the exerted urine with chloroionides and sulfates. Some chloroxylenol monographs likes its pharmacokinetic profile to that of another antiseptic which is rapid excreted in the urine also as metabolite as observed in the human model. Moreover, in one human subject administered 5 mg intragulately, 40% was excreted with glutaracin acid and 70% with sulfuric acid at 3 days. Any chloroxylenol absorbed into the body is likely extensively metabolized by the liver and rapidly exerted, mainly in the urine as sulfate and glucuronide conjugates. Rate of elimination. The major route of excretion is like in urine, although some amounts may be found in bile and traces in exhaled hair. Drug oxidation conjunction. Phase 1 and phase 2. First metabolites and phase 2 stable adducts. These are conjunction process. Metabolites polar species and stable adducts non-polar species. In the form of polar species, renal elimination urine. In the form of non-polar species, biliary elimination stool. Clearance. The only data available recording the clearance of chloroxylenol is the mean clearance rate of 13.76 liter following a 200 mg intravenous single dose of the substance into healthy mongrel dog subjects. Moreover, in another study, when 80 gram of chloroxylenol was administered dermal on a human subject in an alcohol glycerine vehicle, 11% was excreted in 48 hours. Toxicity. As a chloroxylenol is predominantly employed as an active ingredient in various liquids or creams as cleaners, disinfectants or antiseptics that are generally designed to be used topically. It is widely accepted that the use of such liquids, when used appropriately, is unlikely to present a sufficient volume that could be ingested to cause medical problems. In the event of accidental eye contact was with lukewarm water. Chloroxylenol is known to have a low systemic toxicity even at dosage levels many times higher than those likely to be absorbed during normal usage of the agent. Toxicity damages. Toxicity endpoints: neurotoxicity, cytotoxicity, reproductive toxicity, oxidative toxicity, dermal toxicity, immune toxicity, ocular toxicity, and carcinogenic toxicity and oxidative stress. This is last my topic. Thank you for listening my presentation.